Dr. Abed Chaudhary to deliver his lecture. Dr. Chaudhary.
Okay, thank you, everyone. I think yesterday went very well, and the main feedback I got from a number of people yesterday that they were very surprised that these are people coming from other field, and they thought genetics was something very technical. But the way I presented, they felt that it was not technical and they could participate in it. So I think genetics is never technical. Genetics was made technical by scholars more recently. The ancient, timeless events that gave rise to what we call genetics is not symbol ridden, it is not mathematical, it's not technical. It's it's like poetry. And the point I tried to make last time is that till Mendel, and I'll introduce Mendel today, but till the time of Mendel, genetics was driven by what we call phenotype in technical language. That means what you see. So black husk rice is black husk rice, red tomato, eggplant, which happens to be white rather than the typical eggplant purple color, etc., etc. So these are like your experience. So you see the red tomato, green tomato, and there's nothing technical about it. It's the experience of your life. This is also genetics. We did not call it genetics. We should call it genetics. And that genetics did not require any kind of symbol of genotype. It was simply what we now technically can call phenotype, which is the subject matter or the property, curly hair, straight hair, color. A person who is black happens to be black skin, or a white skin, or yellow skin, so on and so forth. So we covered a lot of ground yesterday, setting the scene for what is known as Mendelism, where around 1880s, I think, around that time, or 1870s, there was a person in Brno, in Czechoslovakia, who was doing some experiments. I may be wrong on the exact city or where it's located, but he was playing around. He was a monk, and he was playing around with the pea colors and the shape of and the height of the pea plant. And we also showed how the pea plant came about, the domestication of pea for thousands of years before Mendel. So let's go to the first slide, please. So we are now entering the kind of the heartland of formal genetics. To most people who go to universities, mostly people who go to university learn genetics. We are starting a new stream where people who never went to university could also learn genetics. But let's come back to the formal structure. And you can see the picture of Mendel and pea plant that he made so popular. So the way we call it the foundational principles of genetics was established by Mendel. And he came up with names such as genes and alleles and established what is known as independent assortment. So we don't have to make it mysterious because now we know everything. And we are doing it through the hindsight, almost like going back and giving some historical explanation. So we don't have to proceed like he did, where in tiny steps he was discovering the principles of genetics. We know the principles of genetics, and the most mother of all genotype is the sequence itself. So we know we can just sequence the gene today. And even after knowing how to sequence the gene and knowing the whole code at the level of base pair ATGC, which is the chemical description of the gene, even after knowing all that, we still want to retrace and study the history of how it all came about. So that's what we are doing. And independent assortment, if I could just tell you one of the big hoo-ha about Mendel's law. Independent assortment means that the trait he chose were in two different chromosomes, and they were segregating independently. So that's what it means. So the two traits he chose were in two chromosomes. If he had chosen them accidentally, which happened to be in the same chromosome next to each other and linked, he would not have got an independent assortment. And I also told you that Mendel studied pea and got all this segregation and independent assortment. And he studied another plant, which is the dandelion, and that did not show Mendelian behavior at all. And that turned out to be apomictic, and Mendel kind of stayed quiet about it. It's only after his death, his notes, and found that he found another plant where genes were not segregating. And it was totally confusing. And now we know because that plant was apomictic. So next, please. So I will not belabor this point. This is a retroactive justification of what independent assortment means. And he chose two different traits. And here you see the character of the chromosome as they would line up. So this is where things get very technical and people get scared and want to run away from genetics. And this is not important at all for the spirit of genetics. These are some kind of technical detail of how chromosome would pair and assort. And my talk is at all not about that, but I still want to cover that ground, show you that what he was observing, and he had no idea about chromosome or anything, but from the algebraic relationship, he was surmising that these traits were assorted independently. So at that abstract level, they were independent traits. But now we know that is because of the height and the color of the P, etc. They were residing in different chromosomes. And as a result, they were segregating independently. And that whole algebraic relationship or number relationship is shown in the next slide. Next slide, please. And y, small y, yellow, and what is not yellow, green. So you can see what turns out to be based on these slides, I think the things, and you can do those kind of algebra. So what it means is that it is the 9:3:1 ratio. If you have two genes assorting independently, then their relation in the box on the right, that what is green and round and yellow and non-round and so on and so forth. And then, so that is what we now call Mendel's law. The two different traits will assort independently in a dihybrid cross. In a monohybrid cross, what it means is that if you cross one trait, so let's say with maize chromosome, and we'll again show a video like that. If you have a white maize, which has a white kernel, and another one next to it is a red kernel, then if you cross it, then what you'll find is that the white will disappear. When the red kernel pollen falls onto the white female, and then you got a kernel, and that kernel will turn red, and the white will be hidden under redness. So white is still there, and then later on white will show up. So this trait of when whiteness is recessive over redness, which is dominant. And you can, it's just a common sense. So if you have a blob of white paint, and in another bucket you have blob of red paint, and if you put the red paint onto white, the whiteness will go away, because we know common experience that redness is dominant over whiteness. Redness will mask the whiteness, but whiteness will not mask the redness. It will become pink or something. So this is just a common experience. This is what dominant and recessive means. That something which makes a pigment is dominant over something that does not make a pigment. Just like a red color is dominant over white color. So that's what dominant and recessive means. Another thing is that, so if you have a white dog and a black dog and you cross them, then the whiteness in the hybrid, the whiteness will go away, simply because the black pigment is being made even in the presence of white pigment, and that will show as black. So this is what Mendel showed in retrospect. Once we go and understand it, it is just common sense. But he also came up with this idea of genotype and phenotype, because he started this convention of calling yellow big Y, and the absence of yellowness, which is green, as small y. So big Y over small y is not very intuitive. So it's kind of doesn't really help in understanding things, in my opinion. But if you want to do a genetics course, then you have to write all these genotypes. And genotype is also something that makes you look like a scholar, because otherwise you look like just a farmer counting seed. And when you start writing big yellow and small yellow, instead of saying green, you say small yellow, which is the absence of yellowness. Then you look like a scholar. So this is just a convention. And one thing I must say, that when Mendel came, this is when genotype and all the scholarliness entered genetics. Because suddenly the big yellow, small yellow kind of people who write this nomenclature, they're not farmers, they're scholars, they were in the laboratories. And farmers kind of got left behind from innovation at the time of Mendelism. And we have followed the tradition of Mendelism and become more and more scholarly till such time that we are now sequencing the genome. And in this exercise of sequencing genome and so on, farmers, the innovative farmers, are not with us. So a disconnect has been created, and I should explicitly point it out. Next slide, please. So now I want to, because Barbara is very popular. I have some photos in a meeting I dug out in 1984 when I was there and Barbara McClintock was there. I'll post it in my LinkedIn. I don't have time to show it today, but maybe I'll show it tomorrow. So she's someone I met many times in 1983, 84, when I often went to Cold Spring Harbor. And I used to stay in a little dormitory building, and in front of the building was the maize field, and she used to come and work in the maize field. She just looked like a farmer working the maize field. And she became very famous because we all knew and we learned from Mendel that the genes are in the chromosome and genes are stable. And she showed that the genes can jump out and go into another place, and that therefore the term jumping gene. So she is known as the jumping gene lady. And jumping gene is also called transposable element, transposons. We will have a lot to say about transposons tomorrow. But I want to give you a flavor, just a flavor of how she arrived at what she did. Next slide, please. So she noticed that in the white kernel of maize, a funny color, blackish color was developing. And you can see on the extreme left, there is a black kernel and the white kernels. And then in the white kernel, the spotted regions were arriving. And nobody understood what that was, because that looks like an instability genetically. And she was so perceptive that she looked into all these chromosomes cytologically, and she looked and looked and looked, and over a long period of time. There's another guy called Marcus Rhodes was working as well, and she had other colleagues. But because she was a woman, and it wasn't very usual for a woman to be in science, even after Marie Curie, even in the Western world, it wasn't traditional for women to be in science. So people didn't take her very seriously when she said that chromosomes were breaking and genes were jumping, etc., etc. To make a long story short, she was right and everyone else was wrong. And she ended up finally to be the sole recipient of Nobel Prize. Often Nobel Prize is shared between different people, but in this case, like Marie Curie, she was the sole recipient. And I just want to give a molecular description of what it is all about. The next slide, please. So imagine on the left, so her system is we now call it SEDS system. So SE is a gene which makes an enzyme. And that enzyme comes into this region called DS, dissociator or dissociation, she calls it. And things jump. So imagine there is a pigment gene and there is a whole jumping gene sitting in the middle of the pigment gene, so that the kernel is now white because that gene is disrupted. And when the jumping gene gets into play through the presence of AC transposase, then the DS jumps out of the gene. And when it jumps out of the gene, the redness, the color kind of comes back in a patchy way. So this is what was happening actually, that the disrupted gene was being disrupted by a jumping gene. And when the jumping gene moved out, then the original color, because it was restoring the gene partially, and the color was showing up. So now we know this is what was happening. And what she said, obviously, and the way she explained it was really outstanding and started the whole field of what we now call transposition or jumping gene or transposase, which is now also being used to make a new kind of gene editing. So she stands like a mile in Bengali we call it mile follow, like a seminal kind of a pivot or a central figure. And similar to her, of course, is Jacob and Monod from France, who discovered the operon idea. And because of her work, this was the first step of talking about gene regulation and the gene jumping out from another gene, which we now take for granted, this kind of activity. But she started the whole thing. Next slide, please. So she got the Nobel Prize, obviously. These are some of the salient features of her life. She eventually got the Nobel Prize. I met her in Cold Spring Harbor before just prior to getting the Nobel Prize. And she even came to my university in University of Oregon. She was invited. She was a very simple lady. You could approach her, talk to her, and have a little chat with her. And she didn't have any arrogance or any kind of powerful sense of who she was or anything. She was very modest. She was very persistent. She was a hard worker. She basically worked like a farmer day in and day out in the maize field. Her impact on genetics was colossal. She was also not an establishment person. She never got any official job or official director or any kind of big job she never got. She continued to work like basically a student, and her work eventually obviously led to what we now call molecular genetics. Next slide, please. So her recognition was delayed. It was the end of her life. She got the recognition. Cold Spring Harbor Laboratory, where we went, I went many times in 83, 84, is really a big center of learning for molecular biology. And tomorrow I'll show you some of the photos which I happened to obtain. She conducted all her pioneering work. It was a residential laboratory. There was living quarters. She could live there, work there in the whole campus. And her work was in maize, as we will introduce maize in this class as well. And the long legacy she has left behind is this relentless work. I learned it from her myself as well, to continuously work, and not try to be a big shot or anything, but just continuously work. And that is a big legacy that she has left behind. Next slide, please. So now I want to give you a sense of the fact that Mendel was not the final story in genetics. There's a guy called Morgan. So in America, at least, they call it Mendel-Morgan genetics, as they should, I think. Because he showed that these genes, Mendel's idea of genes, were located in chromosome. He used fruit fly as a model system and made the first genetic map of the fruit fly. He also showed that genes could be linked, so they're not assorting independently. They could be right next to each other. He introduced the concept of linkage. And he also showed that using mutagen, ethyl methane sulfonate, and various chemicals, small molecules, you could mutate the genes, mutate the nucleotides from one nucleotide to another, and therefore you could create genetic diversity deliberately by mutation, as I will show here through my own work. And that whole procedure of whether you study the existing variants of P or whether you directly create mutation is called forward genetics. So this is very important because later I'll talk about reverse genetics. So anything that exists in the world, yellow pea, green pea, there's kind of variation, or, tall pea plant, short pea plant, these variation. These are all manifestation of what we call the forward mutation, because something has happened in a gene and now you see the result. And that forward mutation can be created by mutagenesis as well. Next slide, please. So Morgan is a landmark person like Barbara McClintock. He got the Nobel Prize in 1933, long time before Barbara. And he popularized Drosophila or the fly as the model organism. A lot of work was done by Morgan and his students, Studevant, and they generated, as I said, linkage map. They put a lot of experimental emphasis on working in the detailed genetics, in the forward mutation kind of thing by making mutation with ethyl methane sulfonate. And what we now call genetics is due to Mendel, but much more so due to Morgan. Next slide, please. So several things we need to talk about now, and now we have come to my own work. Because, I want to give you a very primary example of what I have done and why am I doing this course is because I have played a very significant role in my own way in doing mutational analysis in a number of important traits in plant biology. And I will give you some example on it. So the first thing I took on was to identify and clone genes which are involved in seed size. As you know, that rice plant and other crops used to be tiny, tiny little originally. And then the seed size, through human participation and selection, seed size got bigger. And it's because seed size got bigger that we have modern agriculture. Because if we have tiny seed, the food production would be very, very low indeed in terms of weight. So I was very aware of it, and I decided to take it on in Arabidopsis, which is tiny little seed, but I looked for even more tiny mutation in the seed so that seed would get even smaller. And I even screened under the microscope. Arabidopsis seed is very small, and I looked for even smaller mutant. Next slide, please. So this is a piece that was written on my work when it was sent out. My paper was sent out to Proceedings of National Academy of Science in USA. They not only did they publish, but they kind of asked one of the biologists to write a review on or kind of like an editorial on our work. And here you see the whole biology as part of what they wrote. Is taken from that paper in Proceedings of National Academy of Science. So the seed formation is a result of double fertilization. A lot of people don't know that. So from the pollen comes two nuclei. One is the sperm, which fertilizes the egg and becomes the embryo, shown in the middle. And the other one fertilizes the endosperm and becomes the triploid, fertilizes the central cell, which is a homodiploid, which fuses, and then another sperm comes from the pollen and it becomes a triploid endosperm. So one way to understand endosperm is when you have coconut juice. And coconut juice is nothing but liquid endosperm, which later becomes very, very hard. And you can scoop it out and have this delicious coconut. And if you open a coconut, you'll see that there is a baby coconut plant is there, which is the embryo. So coconut is a good way to understand the relationship between endosperm and embryo. And in our case, of course, the endosperm is transient, and you can see in the right that endosperm disappears. And then you have a seed when upon planting, you get a seedling to come out. Next slide, please. So there is a crosstalk between maternal tissue, zygotic tissue, which is the embryo, and a lot of growth regulators play an important role in it, such as cytokinin, gibberellin, etc. And then environmental factor, of course, the soil quality. Agronomic importance is that, of course, seed is what we eat. Seed is also what we plant for the next generation, which is the whole backbone of agriculture. Next slide, please. Next slide, please. So we cloned we identified several genes. The mini seed, we call it mini. Haiku, haiku is a very small Japanese poem. So one of our collaborators called it haiku for indicate smallness. And then we studied it, obviously, and we found some gene regulation, some important genes like kinase, and these are issues, and Warkey transcription factors. These are issues that I can't really get into, but these are the identity of the gene that was defined by the mutation that I generated. along with my colleague Ming Luo. And this was the first mutational analysis of seed size. So that is one of our contribution, I should say, me and my postdoc and my technicians and so on. We did a mutational analysis and we isolated the seed size genes. And these seed size genes became very important later on through work of subsequent people. So this is what happens in science, that I did this work, I left it, and then other people came into the field and then developed it to the very high level. So that is the one thing we did. The second thing I did is isolate a mutant called AMP1, which is codes for a glutamyl carboxypeptidase. Again, it's a very important gene in plant biology. Next slide, please. And you can see this from my paper, that it shifts the Arabidopsis plant shown here on the left in the bottom picture. And on the right, due to a single mutation, you see what happens. It becomes extremely bushy. And it has a higher rate of leaf initiation. As you can see, on the left is a normal plant, and on the right is the AMP1 plant. So it's making a lot of leaf very quickly. Next slide, please. And then it shows an extraordinary phenotype, as you can see the lower right, where instead of 2 cotyledon, there are 3 cotyledon. And normally in Arabidopsis, as you can show in the top, there are 2 cotyledons and leaves comes in 2 pair. And what I have created now is that leaves, cotyledon is 3 and leaves are also 3. So that the whole symmetry has now changed and leaves are coming in 3s rather than 2s. Sometimes there is only 1 cotyledon on the left. So this mutation broke the nexus between monocot, dicot, etc., which is very old in botany. But the presence of my mutation showed that this dicot, tricot is not really a useful definition, because here is a plant which was actually not dicot but tricot. So how do you do with it? And it's even some of the seed are even monocot, as shown in the left. So the monocot-dicot divide was not useful, as the AMP1 mutant showed. And with the help of one of my colleagues in CSIRO, Chris Halliwell, that Diana knows, that we later cloned AMP1. Next slide, please. So other thing I found that if you have AMP1 mutation, then you have a lot of callus and a lot of regeneration. And this AMP1 mutant, instead of dying after 6 weeks, Arabidopsis, as is a normal Arabidopsis, 6 to 8 weeks life cycle. And this one seemed to go on forever, even 6 months or a year or something. And so essentially it had become perennial. And that was, this I did so many years ago. This paper was published in 1993, as soon as I came to CSIRO, because I got this mutant with me from America. But it gave me the idea that plant could be made perennial. And this mutant was due to upregulation of cytokinin, which taught me that by increasing cytokinin, you can make annual plant into perennial, which was like my stepping stone to what happened later in getting perennial rice. Next slide, please. So cytokinin level was high. Embryonic pattern, as I showed you, instead of dicot, it was a tricot and tetracot even. It was a vegetative growth was completely altered, and photomorphogenesis is something that behavior in the darkness is another issue. Next slide, please. I think these are some of the litany of phenotype of AMP1, which is not particularly relevant here, but the seeds, everything was affected, basically. Next slide. So now what I have shown here is that I have shown example after introducing Mendel. I have shown example of my own work where I have mutagenized and got forward mutation in 3 different characters, namely AMP1, which changes the annuality of Arabidopsis into perenniality, or 6 weeks life cycle into the annual life cycle, and was due to the plant hormone cytokinin. And I have shown example of how mutational analysis can be done by forward mutation to alter the seed size and thereby clone genes important for seed size, which is a powerful agronomic trait. I have now come to the transition point where I introduce my own area where I do research, which is Kanihati. And you can see drone images of this lush countryside where I work. And very soon I will introduce you to that area in the work. But I think I'll take a brief break here and show you a video. Mushtaq, do you have the video?
Sir, I will show the video.
Okay. Yeah. We can't hear the sound, I think.
সামিদ সাউন্ড অসুবিধা হচ্ছে?
আচ্ছা, আমি মাইক্রোফোন কি অন করে রাখবো?
হ্যাঁ, মাইক্রোফোন অন করো।
আচ্ছা, ঠিক আছে।
তাও আসছে না বোধহয়, না?
আসছে না।
ওকে,
লাস্ট টাইম তো হয়েছিল।
আচ্ছা, আমি একটু ট্রাই করি। হ্যাঁ,
ওকে, শিওর।
ওকে, ঠিক আছে, আই এম ট্রাইং। Give me a few minutes, probably like 1 minute.
So I'm here. Sharing this. There. Education in biology, which is more related to experiments. And basically what we want to do is to go back. Can we all hear?
Yeah, we can hear now. You can increase the sound a bit, maybe here.
Introduce from the very beginning to students in the natural world, and in particular experimental organism, or what sometimes we call model organism. So there are so many things in nature. If you look around it, it's a bewildering array. There's a mango tree, there's a palm tree, there's a coconut tree, there are elephants, there are tigers, so on. But how do we actually find from it the essential messages, the message of biology, the mechanism? And this is kind of going back to very old-fashioned biology, going back to the time of Darwin or Mendel, and introducing the concepts of variation, which I think our students have really moved away from it, and they don't have a good grasp of it. So when we look at a model organism and what has been tried before, the most common thing, of course, around us in Bangladesh is rice. And here is a panicle of rice after ripening. And this is sort of what you see that farmers cut, and then that goes through the process of processing, first extracting the grains from these panicles, and then removing the husk and getting what we call the actual rice or chal rice. So this is what we cook and we eat. So the difficult thing is that rice is really not the best organism to study genetics, especially at the school level or someone who is getting introduced to genetics, because of the way the flowering occurs, and the self-pollination is very rapid, and it's comparatively more difficult to do across in rice. So we can't just introduce rice into a school and expect people to learn genetics very rapidly. In contrast, although historically it has not been our crop, maize or corn, we call it bhutta in Bangla, has been grown even in this country for many, many years just to produce the corn and eat it. But in the US, in coming from Central and South America and later on in America, corn has been used historically in the departments of biology, genetics, etc. to teach genetics. It's very convenient for the obvious reason. Here you can see there is a abundant genetic variation in kernel color. So these are white corn, and pollen has fallen from a neighboring yellow corn variety, so producing the hybrid, which is yellow. And here you can see this is available even in Bangladesh among our tribal or aboriginal people, the Chakma people and the Tripura people and so on. Adivashi, we call it in Bangladesh. And they have been custodians of this genetic diversity in corn. And you can see this red color, and in fact, a lot of little variation, even within red, suggesting of different modifying genes. So we can use this material to teach basic genetics very easily. So if we take a white corn, for instance, it will produce white corn through generation. And then when we put a yellow or a white one next to a red one, we start to get this red dots, which is the red kernel in the maternal body of the cob, which is essentially pale yellow. So it's very clear, for instance, that the pollen of red variety growing nearby has landed on the silk here. The silk used to be here, and producing these are hybrid. And we can draw them up and show that they segregate 3 to 1. So, but what is important to notice is that these color genes, which are essentially a very important tool for teaching in color. So maize used to be red, and even rice, as you can see here, we developed and collected varieties of rice which are red, different red, and this is almost black and white, just to make the point that all rice is not white. Traditionally, rice used to be red and even very dark red and black, the traditional variety. But the problem with rice is that because of the husk, we cannot see the color inside. So we cannot use it as very essential or easy teaching tool in rice because of the husk. You have to remove the husk to see the color. But maize has the naked kernel. There's no husk outside. So corn or maize can be used for teaching. So here in Kanihati, which is a village in eastern Bangladesh, we have started a project or some process of teaching basic biology, which we hope would be useful not only for Bangladesh, but the rest of the world as well, which is to introduce these basic concepts of biology using very simple material. And we are sitting in an area which is surrounded by rice fields. So obviously we come back to rice, but we have introduced corn in a big way, the colored corn. And what we want to do is to train the teachers, train the local community people, and say that, look, corn is something you can eat. There's a variety we grow is popcorn and sweet corn, and baby corn, etc. So these are all material that people can eat, but they can also learn very easily using the color trait, the unique characteristics of naked kernel and the color. And starting from very basic genetics that Mendel worked out for us many, many years ago. And going back to modifier genes, going back to the concepts such as dominant, codominant, as well as transposable elements, the kind of work that Barbara McClintock did using the kernel color. So all this is possible. And what has been happening not only in Bangladesh, but a lot of what we call developing world, is that the excessive bookishness, the learning, students are learning or forced to learn everything, memorizing a book, and they're giving exam in that bookish learning. And they're not getting even the total intellectual grasp of the fundamental issues of biology, such as variation, what is dominant, what is recessive, basic Mendelian genetics, basic transposable element kind of genetics. And all this is possible to introduce through this informal process whereby we show more experimental material rather than books. So this is kind of a beginning in that direction. And we hope in coming days and coming months, we can introduce more and more concepts of biology and introduce it in a way that the students and lay person who want to learn genetics and biology slowly can grasp the essentials of biology in this.
Thank you very much. So as you can see, I was basically giving this lecture maybe 10 or 15 years ago. Basically, I basically taped what I said today, and this is a very old video, as you can see that I have moved on and look a bit different now. But essentially my point is that we should throw away books and learn from nature. And that is how we have a very powerful learning. So anyway, I had a bit of a break, and now I want to kind of show you something very similar, I guess, in a different way. So this is my village, and you can see even my little ponds and my home, and we have a railway station where the train is arriving. So it's a very scenic scenario, so to speak. Next slide, please. This is a drone photo, and on the left you can see the little Palki River, and on a distance you see the Manu River, looking like a horseshoe, and the paddock that you see, the rice field that you see in the middle, and indicating PV, which is Panchabrihi, is my experimental field. So this is just to give you an example that I don't have a little building with a backyard where a research institute has some land. This is a very large land, farmer's land, where I actually directly do the research on the field itself. I don't do any research in a building, in a laboratory, or in a backyard, but in a very large field. Next slide, please. And what we do, obviously, corn is something I showed you before, but these are the real corn from our field. You can see the corn plant, and on the right is a red corn. And we play around with color a lot because every color is so you take a primary color like red, and then every different color that you get is a modified gene segregating. So it's a good way of getting interacting gene, modifying gene, which is basically affecting a primary color. So this is a good way to getting into the lot of detailed genetics, simply by looking into the color on the kernel, as shown on the left. And you can see infinite number of variations that you can create just by simple genetic crosses. So these are not GMO, these are not CRISPR-Cas9. There is nothing modern about it. It's all classical, old-fashioned Mendelian genetics, or even pre-Mendelian genetics. Next slide, please. Oh, I want to introduce you something very recent that I have done. And in order to understand it, you pay close attention to these two plants. Look at the one on the left, and from which a first harvest has taken place, and you can see the cut region on the top. And after that, only small, mingy little plants have emerged. So this is called a ratoon. This is very poor. Rice normally does not give a second harvest after planting and then withers away. To make a long story short, I looked for forward mutation and also looked by breeding and trying to get something on the right, which is upon cutting, almost like a bouquet of flower, you get the second harvest. So just like my other forward mutation that I've shown you, EMP1, seed size, etc., you can think of my another project where I've looked for forward mutation, whereby rice is being forced to give a second harvest, as you are seeing here on the right, on the picture. It's very black and white. It's a new trait that rice has attained, that upon cutting, it gives a second harvest. Next slide, please. And when we look into the second harvest and that whole biology, third harvest, and so on, we find that they make immense root underneath. So here you're looking on the right, 3 on the left from the farmer's field, which is a typical rice root. And look at the root that I have generated. We dried it and took the weight of it. So it goes from 8 grams to 101 gram, so to speak. Yeah. So more than 10 times. And just to excite you a bit, I'm showing you this picture from the from internet where somebody is showing how big root can be. So we haven't made it that big, but we are getting there. Next slide, please. This is another example where seen very clearly. On the left, you see what is the normal weak ratoon is on the leftmost farmer's variety. And then look at my 3 plants on the right, where upon cutting, you get a second harvest. And then after subsequent cutting, it will be third and fourth and so on. And you can already see that how big the root is of these plants on the right compared to the one on the farmer's variety. So it is massive growth above the soil, massive growth below the soil. And these different Panchabrihi type have different phenotypes. The first one on the left is very strong, and the second one is not so strong. Third one is still weaker. So you get a variation just like you in anything. Next slide, please. And this is to give you an example of how we do it. So it is not some kind of very traditional stuff. We do 3 replicates. We phenotype it from the drone, as you can see on the left. All the varieties are planted in 3 replicates, random block design, and we fly our drone and we measure whether the flowering has taken place or not. And on the ground, it looks pretty much like on the right. I think I at this point, I should give a breathing space and maybe 5 minutes break, not so much break, but invite people to ask some questions, as opposed to yesterday when I breathlessly kind of surged on and all the questions came toward the end. Can we have a question time right now? And I'm sure you guys are bubbling with questions or comments, so can we just start that one? Yeah, Professor Pallav, yeah.
Please unmute. Yes.
Yes.
Yeah.
Can you hear me?
Yeah, I can hear you, please. Yeah.
I have a question regarding the Mendelian genetics.
Yeah.
There are first filial generation, then there is second filial generation, and then there is third filial generation. If we have the result of first filial generation, can we predict the result of second filial generation?
You mean if you start with maybe make a bit clear. Let's say we have a trait and we make a cross. This is typically how it starts, right?
Right.
So let's say we discuss it in the context of color. You have a red kernel, and you cross it with the white, and the resulting F1 is a red, right? So the typical Mendelian experiment is that red one you now you don't cross anymore, right? So you have a self-fertilizing situation where you have a red, but the red is hiding the white in there. So if you were to write the genotype, it will be big red over small red, right? So that the process of making the hybrid, you can call it the first filial generation, right? So you have crossed with P1 and P2, 2 parents, and you got the F. And we will arbitrarily decide that the F is a case of red color, just to exemplify for one monohybrid cross. And you have a red kernel which you plant, and that it grows up, and then it pollinates itself. And now you would get 3-to-1 ratio in the second filial generation. You would get 3 red and 1 white. So and then the only other bit about Mendelian is that if you then also say that you have additional character, which is a short plant and a tall plant, then that shortness and tallness will segregate also 3-to-1, but independent of the color. And so you can see just by describing like a story like that, you have a complete description of Mendelian genetics. And you can write it formally in the form of algebra, and you can say length one code for length, one code for kernel color, and you can do all this arithmetic. But the way I understand and the way I want to convey to people is this story, so to speak.
Right. There is one more question.
Yeah.
We normally get black rice. It is not exactly black. It is maroon.
Yeah, it's intensely purple.
It
Intensely
is
purple.
intensely purple. And we find that nutritionally, black rice is better. It has more protein
Yeah.
than the usual rice.
Hmm.
Now, is it through forward mutation or through some other method?
No, it's not forward mutation. So because the black rice is the progenitor of white rice. We used to have black and red rice before white rice came along. So it is by forward mutating the black rice that we have gotten the white rice, right? It is not by mutating the white rice that we got black rice, because black rice was what is called wild type. It was the pre-existing primitive form, red rice, purple rice, etc. And you can imagine that the pigment gene has been mutated, and then now pigment is not being made, and the rice has become white. So that is why what you just said makes sense. By making white, you have lost the nutrient. You have mutated the nutrient away in the process of making white rice.
Right. And
Okay.
Ani, there's one more thing which I am personally interested. Is there like rice is a very water intensive crop. Now, Punjab and there are other places where India is facing severe problems. Soon we will be facing Bengal also. The rice bowls district are Bardhaman and Birbhum. These are the two districts in Bengal. Now, is there any way of having a rice that uses less water?
Yeah, so that's a very, as you can see in my own photo, that rice is sitting on water. So there are all these efforts. This is very difficult to do because you have to create a kind of a dry condition, in an experimentally dry condition and so on. So I myself did not go into this rather difficult line of research, but a lot of people are doing. Often it's GMO, often expressing this, that, or the other gene, and try to make it less drought tolerant, if you call it, needing less water. So there's a lot of research going on, but I have concentrated on other traits of rice, namely the color traits, and then now the.
I liked the idea of having a second harvest and a third harvest, because probably that way the farmer saves a lot.
Yeah.
I mean, every time after the harvest, a farmer has to prepare his land again. And particularly in Punjab, there is a problem of burning the ends, the ends left in the field. And every time, and it creates huge pollution. So, particularly in the city of Delhi. So in Hindi, they call it parali, that is the end of the plant that is left behind after harvesting. So at least once in a year, sometime in a proper time, they can dispose it off. Right. I found that interesting.
Yeah, so if you could connect me with those people, because I would like to offer it as a solution to all this stubble burning. In English, they call it stubble burning,
yes,
which creates the highest pollution in Delhi at a certain time of the year. So I would like to make this rice available. So instead of burning the stubble to plant wheat, they can have a very powerful second rice from there.
Exactly.
So I need some introduction through you. To this
Now,
video.
obviously,
Okay, other question, please. Yes, Sadia.
Hello,
Yes, go ahead, Sadia. Go ahead.
hello everyone. Okay. So I'm very excited to get a chance to attend this studies about Panchagrihi rice. It has become quite a celebrity variety in Bangladesh. So I want to, do we have any test that reacts to the saltwater from area?
Say that again. Sorry. Say that again.
Your network may be disturbing.
Yeah,
Yeah.
you are breaking
water
up. actually.
tolerance it
Yes,
has.
yes, yes.
Yeah, because uh, in the heavy root design, it just came to my mind that it will also be very beneficial where. So sorry, I not audible, right?
Yeah, now it's better. Yeah, now you
Okay,
can
okay.
speak. Yeah.
So my question was, my question was that how saline water tolerance it has. Like, do we have any research on that yet, or like it just came to mind after seeing the root, the heavy root design it has? It might be beneficial for the fields where the soil erosion is. Yeah. So the soil erosion is.
Yeah, I think I can answer your question. We can't hear you properly because of network issues. But
soil erosion rate is high. So
no, soil erosion. No, instead because of more root, soil erosion is not high. Soil erosion is very good. Because of the massive amount of root, there is more carbon in the soil, and the structure of the soil is better. Yeah. So,
Yes, Sunil from India.
okay,
Also.
Sunil, can you speak? Sadia, I think your network is not good. So, Sunil, can you
Hello,
speak? Yes, Sunil. Yes.
am I audible?
Yes.
Sir, actually, I want to know using genetics, how can we deal with climate change?
What exactly do you mean? How do you want to deal with climate change?
Like more trees can absorb more carbon. So with a less period of time, how can we do it?
Yeah, so one of the thing is that what you looked at, that it has more root, which means it has taken more carbon into the soil. So this rice, the Panchabrihi multi-harvest rice, can deal with climate change that way, but by having more root in the soil, and therefore more carbon in the soil.
Right, sir.
Okay, next question.
Mr. Mushfiqur Hassan, please.
Assalamu alaikum, sir.
Yes.
As a student of class 9, I have a question that we have seen that every time the combination of daughter cell is 3:1 in monohybrid cross. Is there any prediction that the probability will change anytime, that 3:1 will not be
No, it will because of the way chromosome segregation occurs, because of the way genetic segregation works. And the only way it will change is that if the trait is really not dominant or other genetic factor will interfere. So let's say if you have a red to white ratio is 3:1. If the redness is being modified by another gene, then the 3:1 ratio will change. So there are situations I can tell you when this ratio will change. But ordinarily, for the purpose of learning genetics, we assume that this ratio will not change.
Can
There
I
are quite a
predict it? Can I predict this earlier?
No, you can't predict it earlier because you don't know whether the modified gene is there or not. You just have to patiently count. And if there is any deviation, then you have to ask why there is deviation. Okay?
Yes, sir. Thank you, sir.
Okay. I think, Sazia, do you want to speak again?
Yes, sir. Sajia asked questions by message. Check message, please.
Okay. So can you read it?
Yes, sir. Is there any research on how much saline water tolerance is that? If we plan to use it as a tool for climate change and implant this variety in coastal belt?
Yes, of course. I have not measured saline tolerance of the variety yet. We should take it to the coastal belt and try the saline tolerance. Yes.
About root system, the root looks very heavy and condensed. Can we use it as a tool to fight against soil erosion?
Yes, the answer is yes. So we should do the soil erosion specific tests. Yes.
Sir, how much irrigation does PV rice require comparing to the other common varieties?
We don't know yet. We should measure the actual water utility. Yeah, usage. Yeah.
So, Mr. Nazmul Haq Roni, please ask your question.
Thank you, sir. Sir, you said you did a second harvest of rice by forward mutation. I think it's a very good idea, sir. But I want to know, is it possible to keep the yield constant in the second harvest?
No, the yield comes down a bit on the second harvest, but goes up again in the third harvest. So the Panchabrihi shows very powerful trait of. Actually, I can probably send some photos that I just got to Tamid, and Tamid, you will be able to show it, right?
Okay. Yes, sir. Yes, sir.
I will just forward something and it will become very clear to people. I just got some photo from Russell. Just hang on a minute. It's a very important question, so I want to address it.
Okay.
It should go to your WhatsApp.
Yes, received one picture, sir.
Yeah. So I can show you a few actually that I can show you another one and show the both of them. Yeah. Yeah. Okay. So show these two.
Okay, I'm showing.
Sir, I have another question.
Yeah,
Is
just
it
wait.
visible?
Just wait a minute. So you
Okay,
can
thank
see that. So you
Is-
can see that after cutting and harvesting once, now you see how after the first harvest, you see this sudden bushiness in the plant, right? So the plant has come to new life and it's growing massively again. So that is why the third harvest become very big because of this. Show the other one, Tami. Next one.
Yes.
Yeah. So you see how these plants are becoming very big again. So this is what you see, a kind of a transition to new growth in Panchabrihi, but not varieties which are not Panchabrihi. What was your other question?
Sir, my other question is, we know there are many WRKY gene.
Yes.
Which of the gene affected seed size, sir? Is it
Yeah,
identified?
so we found some Warkey transcription factor which affects the seed size. If you go read our paper, if you write my name and PNS and seed size, you will see the Warkey and Warkey transcription factor and some kinases, which are in fact in the seed size pathway. Beyond that, I don't have time to go into all the detail, but you can see it's all in the paper.
Okay, sir, I will find it.
Now,
I think Aki,
yes,
Aki, can you speak up? Yeah.
yes.
Yes, thank you very much. Am I audible?
Yes,
Yes, yeah.
Thank you for the opportunity. And as I have done genetics. I have done not much about genetics or read much about genetics, but I had a course once in soil science where I learned about the soil erosion and losing soil nutrient for cultivating same crop or repeatedly cultivating in a field can cause soil erosion or nutrient loss, or soil can a field can lose overall its fertility. So making the Ponchobrihi paddy, making it perennial from annual crop, will it damage the soil quality overall, or will it be overall beneficial for farmers, or how can farmers handle a crop for 3 to 4 years in a land, whereas he can cultivate around 4 to 5 crops in 3 years in the same land? And what about the fertilizer and other maintenance that farmers should follow? I am just curious to know about this. Thank you very much.
Yeah, it will not be 3 to 4 years. It's just 1 year, 3 to 4 times. Farmers don't have to do five harvests. They can do only one additional 1 or 2, and then do other crops. So I think Panchobrihi does not mean that they have to do 5 times. Even the second and third crop is very high. So I think in reality, farmers will want to plant potato or mustard or something, and will not want to do only rice for the whole year. But I'm just giving them option that if you want to do it, they can do it. That's all.
Like, does that mean that Ponchobrihi is annual crop, but will give its crops for 3 to 5 times a year?
It has the capacity to give 3 to 5 times per year, but it does not have to give 5 times a year. It can give only 2 or 3 crops, and then farmers can plant something else. Okay.
Oh, okay. I didn't know that. This is much helpful for farmers, I think.
Yes.
Thank you very much.
Thank you. Thank you. There are
Omar
2 more
Ahmed.
questions.
Omar Ahmed,
yeah.
please speak up.
Thank you, Dr. Abed, for lecturing today. But I have 2 questions. First question about Mendel genetics. So I inquiry about how the deviation in Mendel genetics explain or illustrate the incomplete dominance, like flowering that have color or something like that. Yeah, and could this first question.
Yeah,
okay.
Yeah, very important question. So incomplete dominance or partial dominance, in real world, a gene is not 100% dominant. It will show epistasis, as called it. So we are not going into all the detail, but for instance, if it is red and white, you may find that some progeny are becoming pink because there is another gene affecting the redness of the red, right? So all these details in real world, all these details come into play. But Mendel pretended that these are not the issue. It is a pure issue, but it is not a pure issue. So when you do the real world cross, as you have seen in my maize cob, there are all these other new colors showing up, right?
Yeah.
So that's why Mendelian genetics is not complete. It's incomplete.
Okay. The second question about the rice, you said we can make 2 harvest or 3 harvests from rice, but I'm inquiring about how plants match flowering time in the second harvest or third harvest to give flowering. How plant in the second
I understand. After the first cutting, it will flower and give harvest in 50 days, right?
Yeah.
And then when you cut again, it will grow up and flower in and give a grain in another 50 days. So it has been planned, it has been selected to be like that, to continuously flower and give harvest.
Yeah. Is this meaning this plant, this a new plant, it's early flowering, not like the normal plants?
Yes,
oh, yeah.
It is early flowering. Yes, of course. It has to be first made early flowering, because without early flowering, you cannot become multi-harvest.
Okay. Yeah. Sorry, I have more questions. Sorry. Yeah, but
sure.
About if we thinking how we can convert annual plants to perennial plants or against perennial plants to annual plants, if we thinking about make a specific mutation in the genes that's responsible about like in any pathway, we can make this like sorry, I will think about my question again and maybe write on chat, because I want to save time for
you
other
can write
people
on
to
the
ask me.
chat.
Sorry, yeah.
Write on the chat, but we are not making any hypothesis about which gene it is. I think it's too premature to do that. It is just a forward selection for perenniality.
Oh, okay. Yeah, I understand. Okay, thank you.
Because if you want to say which gene you want to do CRISPR or something, I'm not sure I'm sure you'll be able to know which one to do. At least I don't. So I want to depend on forward mutation.
Okay, but this mutation, it's natural mutation, not a specific mutation. We don't use
no. It is by forward mutation, you mean that you don't know what it is, the mutation. You just look for the phenotype. Yeah.
Oh yeah. Okay. Thank you.
Hello.
I understand.
Saurabh Dev, please speak up. Saurabh.
Hello.
Sorry. Hello, unmute, please. Sorry. Hello.
Hello, sir.
Yes.
I am
go
from
ahead. Yes.
I am from Sylhet Agricultural University, and I have 2 questions about Panchabrihi.
Yes.
As we know, rice cultivation need more water, and for second harvest, I think it most probably need supply for water about 6 months. So, is it possible to supply water for 6 months in rice field? And which area this rice variety is suitable for? I think it is suitable for a Howrah area. What about your opinion, sir?
Well, my area is not Howrah area, but we have year-round water supply. I have either the river or the groundwater. So I have no problem of water in my field. In my field, there is no problem of water, but I cannot comment on. So you can see that I have river, and when I don't have river, I have groundwater. So I have set it up in a way that I have water, right? But every place is different, and I cannot predict about somebody else's situation. Right?
Okay, sir.
Yeah. Okay.
Thank you, sir.
Okay, thank you very much. So I think we want to go back to the talk. It was very nice to get this nice set of questions. So I think this was the last slide. So next slide, please, Tamit. Yeah, so you can see the condition of the field where on the right, you can see some have been cut and it's growing back again. On the left, you see have just been cut very recently and others are being cut. So these are small experimental trial plots that we can grow different varieties and assess their phenotype. Next slide. So as you can see, the PV, the Panchabrihi has started to grow very rapidly after cutting. On the left, the random variety is not growing at all, but Panchabrihi has started to grow very quickly. Next slide. And basically the BR48, one of the varieties, after 3 cut, I think it completely stops growing. But look at the Panchabrihi on the right, how strongly it grows, even after 3 cutting. Next slide. And the Panchabrihi also, the panicle is very strong. A lot of grains per panicle. Yeah, next slide. Now I want to show you some sense of the perennialization program in other crops. Next slide. So we are attempting to do it in barley and oats. Next slide. And there's a quinoa, and there's a local Bangladeshi quinoa called Batua. We are setting in motion programs to make perennial quinoa. Next slide. And the pearl millet here is not very common in Bangladesh, but we collected the seed and we are growing pearl millet. Yeah. Foxtail millet, many of you, I don't know, may be familiar. In India, it's very common. Proso millet, foxtail millet, finger millet, etc. Next slide. You can see the foxtail, and you can see on the right our experiment when we start to get second crop from the foxtail millet. Yeah. Next slide. Finger millet is very interesting because the inflorescence looks like a fist. Ancient people selected it like that. We grow our finger. We are actually attempting in each one of them to set in motion this process of multi-harvest. Yeah, next slide. In wheat, we are trying. Yeah, next slide. Sorghum is very prone to giving multiple harvests. And in case of sorghum, we are trying to extend it to next, even giving more so that it becomes completely perennial. Yeah, next slide. You can see on the left okra, we have gotten the first harvest from the okra from the top, and the top is cut. And now you can see that in this particular variety, okra is growing from the side and giving a second harvest and continues to give harvest. So this is very good for small farmers when they have a continuous supply of okra. So their okra plant, instead of dying, keeps growing. On the right, you can see flax, or we call it tishi in Bangla, which is very nice flax flower. Next slide, please. And not only we got grew flax, and we isolated some fiber, which used to be known as linen in the old days, because another name of flax is linium. So we can actually get the fiber, as you can see here. Next slide. That's it. So I think this has been a long journey. We started with Mendelian genetics, covered Barbara McClintock, proper genetics. Then I showed you some example of what we call forward mutation. So remember this forward mutation. Forward mutation means that you don't know what you're mutating. You mutagenize and you look for a phenotype. So you don't make any assumption of which gene you're mutating. These days, because of CRISPR and gene technology, every student has become mindset into thinking that they have to mutate a particular gene. But you don't have to mutate a particular gene. The whole logic of mutagenesis is that you mutagenize the whole genome and you look for a phenotype. This is the whole principle of forward mutation. So I showed you a very good example of forward mutation, my own published paper, tackling seed size, very important trait, and then tackling a gene called AMP1, which gave me the idea of perenniality, annual becoming perennial. And then I executed it in rice, getting 5 harvest rice. And then I showed you example of all the other crops that we are attempting to make perennial through this forward mutation strategy. So I will stop here and take more questions. I think it has been a very intense sort of session today, covering a lot of areas. And tomorrow we will cover what we call reverse genetics, which is modern genetics, taking a gene and mutating with CRISPR and homologous recombination. And the whole issue of molecular biology is what we will cover tomorrow. But for now, if there are additional questions, we should discuss it or comments. And I would also encourage each one of you, those of you who are in LinkedIn, to write about this course, whatever your honest opinion is. Cite our course and write about it, because we want well-written pieces of our activity in LinkedIn from the students and from the observers and from the experts who have joined us. Okay, more questions? The ones who did not ask questions or speak or spoke, can you please speak up?
Mushfiqur Hassan Siam, please.
Yes, sir. Thank you, sir, again giving me the opportunity to ask my question. My question was that we know that the ATGC is in the DNA and AUGC is in the RNA. Why is different in U and T? What is
Well,
the reason?
Well, I don't know of any reason. This is exactly how it is in nature, and that's how it was discovered. When people discovered DNA, which is the backbone of the genome, and DNA is much more stable, and RNA is very unstable. So RNA has its unique character, and it is not identical to DNA, because RNA is the messenger. RNA is to make tRNA. RNA, there are special RNA organisms, RNA genome organisms. So it is very difficult in biology. If you ask why something is like that, often the why question is not even asked. So I particularly don't know the reason as to why RNA is the way it is. But maybe somebody would know. You can Google it, and some scholar somewhere has probably commented on it. But I cannot comment on it because I don't know.
Okay, sir. And I have another question of Morgan's experiment, that he has discovered that the relative distance of the genes in the chromosome, and in a same chromosome, the distantly gene separate very fastly, but the closed genes separate a little. Why this occurs?
I didn't understand your question. Can you repeat it?
Yes, sir. Yeah, in the same chromosome, the distantly genes separate very fast, but the closed genes separate a little in the same chromosome.
Yeah, but I guess you mean segregate. So if the gene is very close to each other, it will not recombine, right?
Yes, sir.
So imagine if a gene is right next to each other, you will not get a recombination between the gene very well, right?
Yes,
so
sir.
The gene that is close together will appear linked in a cross, right? And if the gene is far away in a chromosome, then there will be a lot of recombination between these two genes. So they will look like unlinked after a certain distance of this linkage is expressed in centimorgan to pay tribute to Morgan. So when the traits are very far away in the chromosome, it will look like they are in two different chromosomes. They will appear unlinked. But if they're very close to each other, then it will look linked. So to give an example, if it is let's say the color of kernel and the height, both genes are very close. So every time you have tall plant, you will also have red color. And every time you have short plant, you will have white color. That means these traits are linked. They will not assort independently. Is that clear?
Yes, sir. Yeah, I have understand. Thank you, sir.
Yeah, it's much better instead of doing it in such a formal way, if you just try to understand in real world what it is. So you can imagine if these two genes if the chromosome is like a string, and these two genes are like a bead, like girls wear the bead in a necklace. So if these two beads are very close to each other, you will not get them to separate because they're so close, right? And if the beads are far away in the string, then there will be a lot of breaking and joining between them. So it's a bit like that. If you visualize it, if you make a model out of it, then you understand it much better than kind of think of it in a symbolic way.
Yes, sir. Thank you, sir.
Sir, one question from Mr. Hassan Farooq. He asked, as Bangladeshi farmers currently cultivate local varieties in 3 growing seasons, will Panchobrihi provide more yields or benefits compared to the existing varieties?
Yes, of course, it will give more, it will give benefit in two different ways. One is that it will give more yield. So instead of, after burro, instead of digging up the soil and planting house, you can get house from the body of the burro. So two crop you can get very quickly, and you don't have to do any digging for house. That will be the most obvious benefit, because you can in fact get 2 house, and then you don't even have to do it anymore. You can plant your own other crops. The second benefit is that it tremendously increases the soil carbon, because it has massive root, and the soil carbon increases. So this is the second benefit. Okay.
I have a question.
Yes.
When I started looking at your work, all that you have explained in last 2 days, what I find, probably at the back of your mind, you have food security, because you are targeting those crops which are subsistence crop, basically millet, sorghum, wheat, rice, of course, maize. And lastly, I saw that you have also got into flax, which is a natural fiber like cotton.
Yeah.
Jute, we have worked a lot on. So probably I don't know if there is any scope of working on jute or not. Here in India, few more natural fibers have started getting used, out of which one of them is fibers from banana plant stem, because they are otherwise also discarded after a year. So those stems are being used to manufacture fibers, and the fibers are robust. I am exposed to it and I am aware of it. It may even be used for, probably Uganda is doing it. There is a company which is doing it from banana fiber, and they are manufacturing good quality of textile. I have a question regarding PT cotton. Has it been done in Bangladesh or somewhere else?
I think BT cotton has been done in India, Pakistan, but to my knowledge, BT cotton has not been done in Bangladesh.
Okay. Because you talked about BT brinjal yesterday.
Yeah, in Bangladesh, BT brinjal was released, so this was a big news. But BT cotton, to my knowledge, has not been released in Bangladesh.
Not been done.
Excuse me, can I interrupt?
Yeah.
Yeah, my name is Mizanur Rahman. I am from University of Sheffield, UK. So I have a little bit of experience working with the Cotton Development Board. And regarding this issue that Dr. Pallab Maitri asking that BT cotton released in Bangladesh with the joint venture of Mahiko of India, There's a CRY-A1 protein inserted into it, and it's already released, and it is now in the farmer's field. So a lot of trial is going on. So yeah, BT cotton is under the farmer's field now.
Okay, okay. Thank you very much. No, I didn't know it because I didn't hear or read anything in many years ago when I was in CSRO in the 89, starting in 88. We Australia developed BT cotton from our laboratories, and Australia, of course, has very strong BT cotton. And in those days, we had a lot of Indians and Pakistanis even come over to CSRO to get trained from different constructs. But subsequently, at that period, I did not see anything happen in Bangladesh. But after that, I lost track, so I did not know. So it's good to know that BT cotton is there.
It is being used in India
because certainly it makes a big impact in use of insecticides. Yeah. So it's a good thing. It's a good GMO because nobody is objecting to it, and it's only fiber. Although some people, because there's a lot of cottonseed oil, so because almost all the cotton in the US and Australia are now BT, transgenic, a lot of people consumers are talking about cottonseed oil not being very good because there is always a consumer resistance against BT, against transgenic crops. So cotton is obviously not food, but cottonseed oil is food. Most of the French fries are made from cottonseed oil. So I think cottonseed oil is GMO. Yeah.
In fact, in Gujarat, both the cottons, the normal cotton which they were growing and the BT cotton, both are grown. The oil mills of Gujarat are using non-BT variety for producing oil.
Yeah, in Australia, you cannot even grow non-BT cotton. It's not legal.
Oh.
Yes. So every cotton that grows in Australia has to be transgenic BT cotton. And I think it's the same way in the US as well.
Okay. But here that liberty is there.
Yeah, India has the liberty. This
liberty.
is one of the because it's a genetics course and GMO is such an important one of the issues, and it doesn't get discussed very widely. One of the issues that a lot of people concern about is that this kind of control will come into play, like you see in the US or Australia, where the non-transgenics are not being allowed to grow at all. So this is one of the objection that people who are against GM crop are saying, that this is a pathway to total control.
Total control.
And I was quite shocked because I have a lot of cotton farmer friends in Australia. So when I heard this news, I first did not believe it because it seemed to me like extraordinary. And the reason is very logical, actually, because people are growing transgenic cotton. And if somebody is allowed to go non-transgenic cotton, then through pollen transfer, the non-transgenic will become transgenic. And the non-transgenic people will get the transgene and its benefit without having to pay anything, right? So in order to protect the business, they banned.
Exactly.
Very, very strange. Yeah. Anyway, no, thank you for the. Are you in Sheffield right now?
Oh, sir, yeah, I'm studying MSc. Though I am an agriculturist, but I am doing my molecular biotechnology, molecular biology,
No, but what I'm saying is, at the moment you are in Sheffield.
Yeah, I'm in the Sheffield now.
Okay, very good. Now, thanks for your comment. It's very interesting.
And we are famous for, I want to mention it, that we are famous for water use efficiency, what some participants asked about that. That if we regulate the stomatal conductance, stomatal related regulation of the stomata. So we are working on Arabidopsis thaliana.
Yeah, this This is very important topic, water use efficiency. A lot of people in Australia, Graham Fowler and some of my friends, they have been been working on water use efficiency for a long time. It's a very important topic in Australia as well. Yeah. Okay, any more question? Mushtaq, maybe you can say a few words. We can wrap up a bit early today because, you know, it has been a exhausting series of, you know, many complex topic to be covered. And I think it will be so difficult for people to digest so many things. You know. Yeah.
Thank you, sir. If anyone, if no one has any other question, then I think we can probably wrap up today. Tomorrow we have one question. Okay, so Diana.
Hello, can you hear me?
Yes, Diana, we can hear you.
So, Abed, thank you for lecturing such late in the evening. I'm an hour earlier than you are, and I'm really getting very tired. I was thinking to make this comment just to kind of let things sediment in terms of what you were saying about using mutations and using forward genetics, because it looked like we got a lot of questions about kind of practical application of crops and so on. So again, today you took me back to my PhD when I was looking at mutations and working with mutants myself. And one thing that appears as a contrast from your mutations that you were finding, you mentioned mini-free and haiku and AMP1 and so on. In my case, I was looking at nodulation genes, right? What are the genes from the legume side, from the plant side, that allow atmospheric nitrogen to be fixed by the plant, right? And my approach was forward genetics, but that doesn't matter. Reverse genetics, but that doesn't matter. And one thing that I could say is that I also found a lot of mutants and genes, and I think in 90% of the cases, though, they turned out to be genes that you could predict that are there, right? But in your case, you found things that people could not say that existed, and you didn't go into FIS phenotype today, which is a lot more complex, and maybe you will go into it tomorrow. But even just the simple one that you had there, the triple cotyledon. So the entire grouping of the plants is monocots and dicots. So that means there are only plants with one cotyledon and only plants with two cotyledons. It's a very strong rule across the board, right? Yet you find something with three cotyledons. So this is something that nobody even could anticipate that it exists, right? So
yeah.
Abed, can I anticipate epigenetics and say the reason why that is, why our two mutagenesis programs are so different in regard to the expected or unexpected outcome? And I think that is, maybe you will agree or disagree with me, I think that is because you were looking into the reproductive phase, and reproductive phase is a hotspot for epigenetic events. And epigenetics is what really gives those I don't know, sorcery like magic, black magic like phenotypes, right? Will you have the chance to will you have the chance to take us through your you know, your phenotypes and things like that?
Yeah,
They are otherworldly, right? They're otherworldly.
Yes.
You see what I'm
Yes,
saying? It's not just something, oh, big seed, small seed, or big leaf, or red leaf, or it's not something that you could predict that is there. It's something that nobody can even imagine exists. That it exists.
Yes.
Is that right?
Yes, I will come to the FIS phenotype in a minute, but even coming back to tricot. So when I did this forward mutation, I had no idea about tricot, tetracot, or anything, but I was simply looking for anything that's interesting. This was my first mutant I isolated. It was in Boston, in MIT biology. Somebody gave me some mutagenous seed, and I got to work right away. And what I found was extraordinary, because cotyledon number is a very powerful trait in the morphology of plant. And me being just an ordinary PhD student or postdoc, early postdoc, could not have gotten this prominence if it was not an unknown forward mutation, because I could not have imagined a gene when mutated will give break down this
is the host now.
monocot dicot thing. So I got phone calls from taxonomists of Germany, very hardcore people, scholars, which I probably, they will not invite me in their club or something, but they were certainly interested in my work simply because I have done forward mutation without any preconceived idea. So that was one case like that. The second case is the face, as you said, we had no idea. I had this weird idea that first you remove the pollen and you mutagenize and you demand seed without any pollen. So it was a straight black and white assumption. And what I ended up getting is a whole pathway of polycomb class gene and the epigenetic regulation and things like that. So both cases exemplifies the power of forward mutation without knowing any detail, because you cannot know any detail in order to predict such outcome. That's why forward mutation, and these days people have forgotten the strength of forward mutation because of CRISPR-Cas9 and this kind of a tsunami of thing coming from Jennifer Doudna and all these people saying that, oh, we're going to CRISPR everything. But just by CRISPRing literally every gene, you are not going to get the kind of progress that you can do with 1/10 the cost of doing forward mutation and being very intelligent about screening the phenotype. I think tomorrow it may be a bit, we have to get a bit organized. So tomorrow we will, before we lapse into molecular biology history and CRISPR-Cas9 and everything, we'll give a rundown on the fish biology and epigenetics. That's a very important topic to cover. I think we will cover epigenetics tomorrow. Yeah.
Excellent. Thank you, Abed.
Okay, I think we can probably wrap up today. Siam, you have raised your hand. Do you have any other question?
Yes, sir.
Okay,
I have a question about codominance. We know that in codominance, the true alleles each affect the phenotype in separate. How this affect?
Yeah, so codominance, let's say, I mean, I have a good case of codominance about which I made a video. I think I have to fish it out for you. So if you were to cross something red and something white, traditionally you expect it will be red. But codominance is if you get pink, right? You get a third color where the whiteness directly affects the redness. That is called codominance, if I want to just give you a real simplistic example. So that has to do with biochemistry. Some phenotype is such that one cannot interfere with the other. But when the interference occurs and the third phenotype is happening in the F1, then that by definition is called codominance. And there are many, many reasons. You can imagine biochemical pathway interference. You can imagine so many interference, which gives rise to codominance. What appears to be codominance is the observable phenotype, but the mechanism behind could be many.
Yes, sir.
Okay, so I think we don't have any other question. So for today, we will wrap up and thank you, Dr. Abed Choudhury, for covering the conventional genetics, the classical genetics, concept of classical genetics, the concept of jumping genes, and the contributions of the scientists who made the difference. Thanks, Diana, and also Professor Paulo, for your contribution to the discussion and also the active participation from students and other participants. Tomorrow, as already been indicated, the next chapter will be gene editing. Not only gene editing, I think gene writing will also be covered there. So more exciting things waiting for tomorrow. And we'll join at the same time, 5 p.m. Bangladesh time. I will send the Zoom link this evening or by tomorrow morning latest. So we hope to see you all tomorrow and more interactive sessions waiting there. Thank you so much, everyone.
Thank you.
Thank you.
Thank you.
Bye.
Bye.